Anti-hair loss. Activity of healthy human scalp-derived Staphylococcus capitis. Your scalp is crawling with bacteria right now. Thousands of them. Different species, different strains, all doing their thing. And here's the thing nobody talks about. Some of those bacteria might actually be saving your hair from falling out. Which means if you're losing hair, your scalp might not be the problem. Your scalp bacteria might be. So what if the solution to baldness isn't some pharmaceutical you swallow, but something that's already supposed to be living on your head? For decades, we've thought about hair loss as a purely biological problem. Your genetics load the gun, your hormones pull the trigger, and your hair follicles either survive or they don't. We've gotten pretty good at pharmaceutical interventions, sure. Minoxidil, finasteride, these work for some people, some of the time. But they're also systemic treatments, meaning they affect your whole body, and they come with side effects that make some people nervous about long-term use. But there's been this growing realization in dermatology that your scalp isn't just skin, it's an ecosystem. It's home to hundreds of microbial species that live in a delicate balance. When that balance tips, when the wrong bacteria overgrow or the right bacteria disappear, your hair suffers. This study emerged from asking a genuinely interesting question. What if we looked at the bacteria living on people with healthier hair and asked what they're doing, right? This research came out of two Korean biotech companies, Kolmar Korea and HK Inno.N. The lead researchers, Yu, Gil, Kim, and their collaborators, published this in April 2026 in the journal Microorganisms. These aren't household names, but they're working at the intersection of dermatology and microbiology in South Korea, which has become a major hub for beauty and skin science research. The study is recent, peer-reviewed, and methodologically solid, which is why it's worth your attention. Here's what they did, and it's actually clever. They took 20 Korean people with genuinely healthy scalps and healthy, thick hair. Not trying to sell you anything, just normal people with good hair genetics and, importantly, good scalp health. They isolated the microbes living on those scalps. Then they zeroed in on one species, Staphylococcus capitis. This is a bacteria that naturally lives on human skin. It's not an infection. It's a resident. Think of it like a housekeeping service that's supposed to be there. They grew this specific bacterial strain, the one they called KMH304, in culture. Then here's the key part. They didn't apply the actual bacteria to cells. They took the liquid the bacteria had been growing in, filtered out the bacteria themselves, and tested just the liquid. That Ferment filtrate. It's like they were asking, what chemicals is this good bacteria producing that might benefit hair growth? They then exposed human hair follicle cells and scalp skin cells to this filtered liquid at various concentrations and measured what happened. So what happened? The cells got healthier and more active. In the hair follicle cells specifically, the Ferment filtrate increased cell density after 48 hours. The effect scaled with concentration. More filtrate, more benefit, up to a point. But it wasn't just raw cell proliferation. The researchers found that the filtrate controlled specific growth factors. We're talking about KGF, IGF-1, and HGF. These are the molecules that tell hair follicles to keep growing. The filtrate optimized their expression. It's not like it cranked them to maximum. It regulated them, which is actually more sophisticated than just stimulating everything. Here's where it gets interesting. The filtrate also suppressed genes related to androgenetic alopecia. That's male and female pattern baldness. Specifically, it reduced androgen receptor expression and TGF-beta2, which are the actual drivers of pattern hair loss in genetically susceptible people. Again, not by nuking them entirely, but by modulating them downward. That's a big difference from a sledgehammer approach. Then the researchers looked at aging. Hair follicles age. They enter senescence. Dead cells accumulate. The team found that the bacterial filtrate activated sirtuins. These are longevity genes, literally called the aging genes, because they extend cellular lifespan in everything from yeast to humans, specifically sirtuins 1 and 7. The filtrate also ramped up collagen production in the hair follicle while suppressing PT1, a marker of cellular aging. Under the microscope, cells treated with the filtrate showed less of something called X-gal staining, which is literally a visual marker of aging. The cells looked younger. In the skin cells, the keratinocytes that form your scalp barrier, the filtrate did something equally important. It boosted filaggrin and involucrin, which are the structural proteins that hold your skin barrier together. Your scalp barrier is your defense against irritation, infection, and inflammation. If that barrier is compromised, everything else falls apart, including hair growth. The filtrate also showed antioxidant activity, meaning it scavenged free radicals. They tested this with actual antioxidant assays, DPPH and ABTS assays, the real deal, not marketing speak. If you're sitting here thinking, okay, but will this actually stop my hair from falling out? That's fair skepticism. In vitro studies, cell culture studies, are a stepping stone, not a destination. We're talking about cells in a petri dish, not a human scalp. The effects are real in the lab, but translation to a living, breathing human is complicated. You'd need to show that applying this filtrate actually penetrates the scalp, that the active compounds survive, that they reach the cells at meaningful concentrations, and that all this translates to visible hair retention over months. That said, what this study does exceptionally well is identify a mechanism. It's not just this stuff helped our cells, it's explaining how, which pathways, which molecular players, which directions they're being pushed. That mechanism is plausible. Growth factors matter for hair. Androgens and TGF-beta matter for hair loss. Senescence matters for hair aging. Scalp barrier function matters for overall scalp health. This isn't magic, it's biology. The practical implications are interesting. If this pans out in human trials, and that's the next step, you're potentially looking at a topical treatment derived from fermented bacterial culture. That's unusual. Most hair loss treatments are either pharmaceutical drugs or vitamins. A probiotic metabolite is a different category. It's potentially less systemic, meaning fewer whole-body effects. It might also be less likely to trigger resistance or adaptation over time, since you're not hitting one pathway with a sledgehammer. Where does this fit in the bigger landscape? There's been a real renaissance in microbiome research over the past decade. We've learned that your gut bacteria influence your immune system, your mood, even your metabolism. Your skin microbiome is getting similar attention now. We know that dysbiosis, imbalance in the microbial community, is linked to acne, eczema, and rosacea. The idea that it's also linked to hair loss is relatively new, but it's gaining traction. What we still don't know: does this specific strain work on every person's scalp? Does it matter what your baseline microbiome looks like? Is the effect additive with other treatments? Could you just apply the live bacteria, or do you need the ferment filtrate specifically? Would this work for androgenetic alopecia, or is it better for other types of hair loss? These are the questions the field should be asking next. The biggest open question: if healthy scalp bacteria produce these beneficial metabolites, why do we lose them? Is it genetics? Diet? Shampooing too much? Environmental factors? Understanding that might be just as important as applying the solution. Turns out, your scalp isn't just skin. It's an ecosystem, and the right residents might be worth keeping around. And they're not done yet.